للناس. So hello everyone, it's my pleasure to welcome you to a new MGBS seminar today, and I am really so pleased to have the chance to introduce our guest speaker, Dr.Meriem Aoun, an assistant professor in Oklahoma State University. Dr. Meriem is originally from Tunisia, where she earned her bachelor in agricultural engineering, University de Sousse, and then she got her master in genetics and biotechnology from the Mediterranean Agronomic Institute of Haiena in Greece. And then she earned her PhD in plant pathology from North Dakota State University, where she worked on rust resistance in wheat. She was a postdoc at Cornell University and Washington State University. She studies the genetics of disease resistance to wheat pathogens. And Dr. Meriem is a key member of the Oklahoma State University Wheat Improvement Team developing disease resistance in wheat varieties. Dr. Meriem, thank you so much for showing up today and to deliver your outstanding research. And you can please start your presentation.
Okay. Hello, everyone. Good afternoon, wherever you are. For today's seminar, I'm going to share with you what I start doing in my program at Oklahoma State University since 2022, and that's to enhance disease resistance in hard winter wheat. So I believe Dr. Salam already introduced me. This is the outline of my talk. I will give you an overview on the most important wheat diseases that we have in Oklahoma. And then the first part of my talk will focus on breeding for resistance to wheat viral diseases. Then on my research, on my team research on to enhance rust resistance in U.S. hard winter wheat. So starting with the importance of wheat. So this is a very important crop in the U.S. And it is growing in 42 states. And in the U.S., we have 6 wheat market classes that are shown here in this map. So the different colors represent the different classes of wheat that we have in the U.S. So the most important class is hard red winter that is growing all over the Great Plain from Texas all the way to North Dakota and Montana. And hard red spring is growing in the northern Great Plain, Minnesota, North Dakota, Montana, and South Dakota. Soft red winter is primarily in the eastern part of the country. Soft white is in the Pacific Northwest, and a little bit of hard white that is in Nebraska, Kansas, Colorado, and a little bit in Idaho. And durum wheat, which is a tetraploid wheat primarily growing in North Dakota and Montana. So and Oklahoma is here, so we grow wheat in mainly in the western part of the state. We don't have much wheat in the eastern part. And my talk will be today focus on this market class, which is hard red winter. So in Oklahoma, we are in a dual purpose system. And what it means that we grow wheat for forage and then for grain production. So we plant wheat usually in October. And that's to allow for forage and for grazing. And then when the crop reaches the first hollow stem, which is in early spring, the cattle are removed from the field, and that's to allow grain production. And here is Oklahoma wheat production from 2013 up to 2022 in million bushels. So Oklahoma is the 4th largest wheat producer in the country. And in some years, we have some drops in production, and that's primarily due to severe drought, like in 2018 and in 2022, and this year as well. So 2022 and 2023 were severe. We had severe drought, and that make the production drop from 115 million metric ton to around 69 million metric ton in 2022. So wheat is facing challenges by a number of abiotic stresses like heat and drought, but also by different diseases of wheat that reduces yield. So here in this table, I am showing you an yield loss estimates in Oklahoma from 2018 up to 2022. So on average, we have 10% reduction in yield, and that's correspond to around $55 million loss in production. So in this table, I am showing you here the main important diseases that we have in the state, in Oklahoma. From 2018 up to 2022, we have the estimated yield loss in percent. So by far, leaf rust and stripe rust are the most damaging diseases. But in years like in dry years, like 2022 and 2023, we don't have any significant damage caused by these rust diseases. Same thing for the other fungal foliar diseases like 10 spot, septoria, leaf spot, or also Stegonospora nodorum blotch, and powdery mildew. In dry years like last year and this year, we are seeing high severity of root rot diseases, both common root rot and Fusarium root rots. And we have on average last year 1% estimated yield loss, and that's statewide. For viral diseases, we have barley yellow dwarf. That's a very common disease here in Oklahoma. And in some cases, like in 2022, we have high severity and incidence of wheat streak mosaic virus. I will be talking more into details about these diseases in future slides. So when I joined OSU in 2022, I joined this amazing team that have different backgrounds, and they are led by Dr. Brett Carver, who is the wheat breeder at OSU. And my part in this team is to help the breeding program develop and release disease-resistant varieties. So here I'm showing you symptoms for those who are not familiar with wheat diseases, leaf rust and stripe rust, 10 spot. That's something we see it, especially now that we moved to a no-till system in Oklahoma. The septoria, Stegonospora, spot blotch, and powdery mildew. And for the viral diseases, these are the symptoms of the most important viruses that we see in Oklahoma: wheat streak mosaic virus, the wheat soil-borne mosaic and wheat spindle streak mosaic virus complex, and barley yellow dwarf. So to help the OSU wheat breeding program, this is basically the pipeline that I follow to enhance disease resistance in wheat. So depending on the disease of interest, that can be any of the diseases that I was showing earlier, I do the evaluation of elite lines, and that's basically the OSU elite lines, or can be any hard winter wheat lines from the Great Plains. And in cases where we don't have the resistance in wheat, like for many of the viral diseases, we need to go to wide relatives of wheat. We do conventional phenotyping, but we start introgressing high-throughput phenotyping, and we start doing this in 2022 and 2023, primarily for barley yellow dwarf. For genetic analysis, we do genotyping, basically based on any SNP-based platform, and that's to allow genome-wide association studies and QTL analysis in biparental populations, and hopefully be able to clone some genes. And that is to develop a molecular marker that we can use in marker-assisted selection. Genomic selection, that's something that I intend to use for resistance that is quantitative, like for Fusarium head blight. Now in Oklahoma, the Fusarium head blight is not that important disease, and that's because of we grow wheat mainly in the western part of the state, which is dry. But we have some places like in eastern part of the country, north or central east part of the country, we have some Fusarium head blight in there. But now with the climate change and also with the different time that when we receive precipitation, that can change the situation. So we have Fusarium head blight in consideration, and that's something that I intend to use genomic selection for. And in case we identify resistant genes that are in wide relatives, for example, or land races, we introgress them into the OSU breeding lines and eventually provide those resistant lines for breeders that will be tested for other traits like yield and quality before a variety is released. So to enhance resistance in the OSU breeding program, basically we do each year we do the evaluation of the different breeding lines from preliminary to the most elite lines, and we do the evaluation both in the greenhouse and the field to these diseases. We do in the greenhouse leaf rust, powdery mildew, tan spot, and spot blotch, and I have 2 nurseries in Stillwater for viruses. The wheat soil-borne mosaic and wheat spindle silk mosaic virus, and for barred yellow dwarf. And the number that you are seeing here are the number of lines that we tested in 2022. And we do this regularly. So each year we have a new breeding lines that are tested for these diseases. So breeding for resistance to viral diseases. So here are the most common viruses that we see in Oklahoma. Wheat streak mosaic virus is primarily in the northwestern part of the state. It's in a region that we call the Panhandle, and that's where breeding for wheat streak mosaic virus is something very important in the breeding program. It is transmitted, this virus is transmitted by the wheat kernel mite that can also transmit other 2 viruses, Triticum mosaic virus and the high plain virus. Barred yellow dwarf is transmitted by aphids, both the green bags and the bird cherry oat aphids. And we have the soil-borne mosaic virus and the wheat spindle streak mosaic virus that are transmitted by a protozoa, the Polymyxia graminis. So for in our nursery, we basically try to do the phenotyping, but also to confirm that we have these viruses, because based on symptoms, sometimes it's confusing to distinguish them apart. We do ELISA to validate the presence of a particular virus, but I have an undergrad intern, Maria, who tried to develop a multiplex PCR, whether it's conventional PCR or qPCR, to distinguish between the different viruses. And that is for us to use to confirm, to validate that we are rating for the correct virus in our nurseries, but also we use it for our extension work for diagnosis of wheat viruses that from samples that we are receiving from growers. So starting with the first disease, wheat streak mosaic virus. This is a picture that was taken last season in 2022 in Oklahoma. So this is in a grower field that is severely infected with wheat streak mosaic virus. So I believe around 150 acres of his field were affected, which is half of his field was severely affected, and the grower, he was forced to basically terminate his crop. So this is how the symptoms looks like. And basically, we see the disease here in the Panhandle area. So this is where breeding for wheat streak mosaic virus is a very important goal for breeding program. What you see here is the vector that transmits the virus, which is wheat kernel mite, which is it's difficult to see without magnification. So. And it is transmitted by wind. So the difficulty for breeding or for managing this disease is that both the virus and the vector, they have a wide host range. So they can affect crops like wheat, but also other crops. And also they can affect several grassy weeds. So that makes them very difficult to manage because, and one of the management strategies that we actually use to manage for this disease is to control volunteer weeds and these grassy weeds in the field. And that's to reduce the population of the wheat kernel mite and then to reduce the risk of infection by wheat streak mosaic virus. But variety selection is an important goal, and that's something that, if available, we would like to use. Like these are variety trials where you can see some distinction between resistant versus susceptible varieties. This is in the Panhandle where this disease is common. But in some years, like in 2022, we have this disease spread far east, and it affected several wheat fields in Oklahoma, including this north central Oklahoma in Kildare, where you can see these varieties that have distinct reactions to the virus, resistant versus susceptible. So the challenge with breeding for wheat-streak mosaic virus is the limited number of genes that we have available for the wheat community. So we are primarily now relying on two genes, WSM1 and WSM2, with WSM1 being the most effective and the most desirable gene, because WSM2, although it provides resistance to wheat-streak mosaic virus, but it basically increases the risk of infection by other corn-transmitted viruses like the triticale mosaic virus. So wheat breeders, they tend to prefer using WSM1 than WSM2. And the most recent one is WSM3. It is not yet present in any of the commercial wheat varieties, so it's still in some breeding lines. And again, those resistant genes are not native to wheat, so we are having them primarily from wheat-wide relatives like tynopyron. And that's, and you know, transferring a resistant gene from wide relatives to wheat, that requires a lot of efforts and a long time to have this resistance available in varieties. So what I have here in bold, those are varieties that we have in Oklahoma, WSM1 or these breeding lines that carry WSM1 or WSM2. So ideally, we would like to have WSM1 and WSM3, and that is because they provide resistance to the other viruses that are transmitted by the corn mite. Also, another limitation of this resistance is that WSM1 and WSM2, they are temperature sensitive, which means that they won't be effective at a temperature that is higher than 75 Fahrenheit or 24 Celsius. So that's another problem. But the newly identified resistant gene WSM3, it can provide resistance above 24 Celsius. Another way to manage this disease is also to have resistance to the wheat corn mite, so to the vector of the wheat-streak mosaic virus. So we have 4 genes, but the most effective one is CMC4. And this is because it is effective against the haplotypes, the different haplotypes of the wheat corn mite, 1 and 2. And again, it is coming from white relatives. We don't have it in wheat. And here I am showing you example of Oklahoma lines that carry CMC4, and those are the resistant varieties that carry CMC4. So ideally, we would like to have WSM1 plus CMC4, and in case when we have WSM3 available, then it will be WSM3 and CMC4, or probably a combination of WSM1, WSM3, and CMC4. Another disease that we have, and it's very common in Oklahoma, is barley yellow dwarf. So the most prevalent race or strain is barley yellow dwarf PAV. And it is transmitted by cereal aphids, and the resistant genes that are available are 1 to 4, but only BDV2 and BDV3 tend to provide that protection against the most common strains, which is BDV or barley yellow dwarf virus PAV. And here are the bird cherry oat aphids that vector this virus. So in Oklahoma, or in Stillwater, I have a nursery for to evaluate the most elite OSU lines to barley yellow dwarf. So here, one of my undergrads helping with the phenotyping of barley yellow dwarf. And here is the distribution of the reaction in this most elite lines from 2022. So only, I would say 13% of the lines were highly resistant to barley yellow dwarf. And by looking at the molecular marker data of these elite lines using CASP marker or competitive allele-specific PCR marker that are linked to BDV1 and BDV2, we found that 4% of the OSU elite lines, they carry BDV2 gene, and 11 of them, they carry BDV1 and BDV2. And one of the most resistant varieties that we have in the OSU breeding program is Uncharted, that shows here an immune reaction compared to another variety from the breeding program called OK Coral, where you can see this the infection by Balliol dwarf. So it has a moderate protection against this virus. The third virus that we test for is wheat soil-borne mosaic and wheat spondylothoracic mosaic. So this is a complex because they both can be transmitted by Polymyxia graminis in the soil. So this is a nursery that I have in Stillwater. And the rows that you see that are yellow in color, those are our susceptible checks. And the rows that you see here, those are breeding lines from OSU, but also we have at this nursery lines that are tested from all over the Great Plain for resistance to this virus complex. With soil-borne mosaic is more damaging, so that's why we try basically to have to select resistant lines to this virus. So you see here we have irrigation, and that's basically to help with the infection of Polymyxia graminis and also with the virus infection. So we do the evaluation here early in the spring, and that's because the virus will transfer from the root to the leaves at low temperature. So in April, that's when we have the temperature starts to rise, and that's when the symptoms will disappear. But we still have see a yield loss, even if the symptoms are not there. So that's why for selection, we try to do this early in the spring when the temperatures are low, and you can see here the clear distinction between susceptible versus resistant lines. So these are the results from 2022 we test lines, preliminary line, intermediate, and the most elite lines. And thanks to this evaluation, we passed from no OSU varieties to be resistant to this virus complex to almost all OSU varieties being resistant. And this resistance is primarily due to the presence of SBM1, which is the locus that we target for resistance to the wheat soil-borne mosaic. So we have a cast markers that we use to do the selection, but also we do the phenotypic selection, just in case we have other loci that are providing resistance to the wheat soil-borne mosaic. So here again, high level of resistance regardless where we are at in the breeding pipeline. We also test, as I mentioned earlier, regional nurseries. Those are lines that are coming from all over the US, hard winter wheat that are coming from both public and private sectors. And we use in the breeding program some lines from Eastern Europe, from Hungary and Romania. And before we use them in breeding, we test them for this virus. And in some years, we have companies that they send their breeding lines to be tested for this virus. So coming now to the last part, which is the enhancing durable rust resistance in hard winter wheat. So I will be talking about the two major diseases that we have in Oklahoma, leaf rust and stripe rust. So leaf rust is a very common disease. We see it in the US and basically everywhere wheat is growing. In the US, this disease has a high diversity in terms of virulence on the leaf rust resistant genes. So on an annual basis, and this is based on the USDA Cereal Disease Lab in Minnesota, where they do this annual survey about the races that we have in the state. We have 30 to 40 races that are present on annually. And we have 83 known leaf rust resistant genes in wheat. And many of them, they are no longer effective against the races that we have. What we know in hard winter wheat are these listed genes, LR21, 24, 26, 37, 39, and 42. Those are seedling-resistant genes or all-stage resistant genes, and LR 34, 46, 68, and 77, those are adult plant-resistant genes. I will be explaining about the difference between these two types of resistance in a coming slide. Stripe rust is another important disease in Oklahoma. But this depends on the year. Like in 2021, this used to be the number one disease in the state. And here is a picture taken by my predecessor, Dr. Bob Hanger, who took this picture of a tractor after a fungicide application, where you see that the tractor is fully covered by these yellow spores of the stripe rust pathogen. So that shows how severe stripe rust was in 2021. So here, coming to the different classes of rust-resistant genes, because I will be talking about these two classes of resistant genes in coming slides. So we have two groups. The first one is called seedling resistance or all-stage resistance. And this is a resistance that is expressed at seedling stage, and it will continue at adult plant stage as well. And it is controlled usually by major genes. So we have a qualitative type of resistance. And it is associated with hypersensitive response. So you see this necrosis or dead tissue surrounding the pashol, and that's to delimit the fungal growth. However, this type of resistance is race-specific and short-lived. So this means that if we use a variety that carry a single seedling-resistant gene, that variety is most likely going to be not resistant in a couple of years. And that's why it's not recommended to release a variety that carry only a single seedling-resistant genes. So another group of resistance is called adult plant resistance. So this type of resistance is quantitative, so it provides partial level of resistance, and it is not associated with hypersensitive response. We see some diverse resistance mechanism, different than the hypersensitive response. And the good thing about this resistance, it provides resistance to multiple races and to multiple pathogens. So basically, we have genes like LR34 that can provide resistance to all different races, and this resistance will be expressed only at adult plant stage. So it can provide resistance to different races of the same pathogen, but also can provide resistance to leaf rust, stripe rust, stem rust, and powdery mildew as well. And that's why it is also considered as a durable type of resistance. So that's why this is considered as one of the primary objectives for breeding for durable rust resistance. But because a single gene cannot provide that good level of protection, we need to have multiple adult plant resistant genes in a variety. Or we can basically pyramid seedling plus adult plant resistant gene to have protection both at seedling stage and adult plant stage. So for the OSU breeding program, we do the evaluation of the lines at seedling stage and at adult plant stage. And here we test the intermediate lines and the elite lines in the breeding pipeline. And this is from 2022 data. So here we have the reaction, so resistant, moderately susceptible, and susceptible. And we have the percent of the line that are showing these different reactions. So you see we have a high level of resistance at seedling stage, but also higher level of resistance at adult plant stage. And this is an indication of the presence of adult plant resistant genes. And actually, this was a primary objective of the OSU wheat breeding program that tend to have or to pyramid multiple adult plant resistant genes. So, but based on molecular marker data and using CASP markers that are diagnostic for these resistant genes, so the green bars, those corresponds to all stage resistance or seedling resistance, and the orange bar, those correspond to adult plant resistance. So for the seedling resistance like LR21, 24, 37, we know they are present, but unfortunately, they are not effective against the races that we have. So the protection that we are seeing at seedling stage must be different than these three resistant genes, because LR42 that should provide a broad spectrum of resistance if it's present in low frequency. So we know that we have other genes, but we still don't know the genetics behind the seedling resistant gene that we have in the OSU breeding line, and that's what I am working on. And we have the adult plant-resistant genes here, like LR34, 46, 68, and 77 that are present in high frequency. And here I am showing you a table of breeding lines from elite breeding line from 2022 that are having more than one resistant genes, and those are adult plant resistant genes. So pyramiding multiple resistant gene can provide that high level of protection because one gene by itself will provide a partial protection. So here we have an example of highly resistant varieties like Duster and Uncharted that carry three combination of adult plant resistant gene, LR34, 46, and 77. And those are the percent of the breeding lines that they carry at least two or more adult plant resistant genes. And this was translated in the most recent release varieties from OSU that are having this high level of protection against leaf rust. And that's primarily due to this pyramiding of adult plant resistant genes together. But also for some varieties, we have also the seedling resistant genes. So that work was primarily focusing on the OSU breeding program. So I wanted to expand this to several breeding programs across the U.S. And we have this national collaboration that is coordinated by the USDA, the U.S. Department of Agriculture, where we have breeding lines coming from different public and private sectors, and they were sent annually to different collaborators for testing to different diseases, agronomic, and different traits. So our part in this program was actually to do the evaluation for the virus, for the wheat soil-borne mosaic virus. But I also use this population because I am a RAS pathologist, so I use this population for evaluation to both leaf rust and stripe rust, because this population was already genotyped by the USDA using a platform called MutRANSeq, or multiplexed restriction amplicon sequencing. So this is a simpler version of GBS. So it's based on two PCR, and then we have sequencing, and then we will get SNP markers that are distributed randomly across the genome. And after filtering, we end up around 10,000 SNP markers that we are planning to use for genetic studies to identify the resistant genes that we have in this germplasm. So here we have three different nurseries. We have the regional, Northern Regional Performance Nursery and Southern Regional Performance Nursery. So this is mainly, it has hard winter wheat lines from the Northern Great Plains, and this is from the Southern Great Plains. And those are the most elite breeding lines that we have in the US. So these have a high chance to be varieties in future. While for the regional germplasm observation nursery, those are lines that can be of interest to the breeding program. So they are not necessarily the most advanced line in the breeding program. And that's why we have a bigger or larger number of lines that are in the argon compared to the NRPN and the SRPN. So these is the principal component analysis based on SNP markers, and where you can see how these lines are related based on principal component analysis. So my master's student, Indira, she's working on the leaf rust project for, so she's evaluating this the SRPN and NRPN, the Northern Regional Performance Nursery and the Southern Regional Performance Nursery. For short, I'm calling them RPN, and the Argonne are the regional observation nurseries. So she has a population around 459, and she's testing them with these 5 races of Vaccinia triticina, or the wheat leaf rust pathogen. So we choose these races because they are the common and the current races that we have in the US. And basically the combination of these 5 letters, that is nothing that this combination of high and low infection type. So high is the gene is susceptible, and low is the gene is resistant. So basically, by looking at this number, we can tell what genes are effective and what genes are not effective. And this is basically what we have in Thatcher wheat differential set. And this is what we use in the USDA survey to identify the races that we have in the state. And this is based on Long and Colmer nomenclature. Dina, she also tested this population in the field, and that's basically to look at any possible adult plant resistant genes that we have that were susceptible at seedling stage. So we have this nursery in Stillwater in 2023. So she has the lines planted here, and after 5 ranges, we have a spreader row. So that's a susceptible variety that we inoculate with the spores from Oklahoma. And we have here this border row, which is also a susceptible variety that we inoculate. And that's to try to increase the disease pressure in the field so that we can have good ratings for leaf rust. And due to the drought, like this year, we were doing irrigation, and that's to help with the infection. So here I'm showing you some of her results for like the phenotyping for the 2 races that were done at seedling stage. So these 2 races are the most dominant races that we have in Oklahoma, MPSD and MPPSD. And you can see that we have some resistant lines, but the distribution is more skewed towards susceptibility. So we have an infection type of 0 to 9, with 0 being the most resistant, 9 being the most susceptible. But when looking at the data at adult plant stage from the field, we can see here that we have more or higher % of line that are showing resistance. So this is an indication that we have adult plant resistance in this germplasm. So Indira, she used the SNP markers that were generated from the mutrans, the MRI-seq, and then she did the GWAS using the phenotypic data that was available. So here so you are seeing here only one association per and per maybe chromosome, and that's and just because simply she was using a B-Link or farm CPU model, which are multi-locus models. So we expect to get a single SNP per locus. So these are the results from the 2 races, and this is from the field. So again, we have some variation, and that depends on the race that we are testing. So those can be race-specific resistance, and also some, for example, associations that appeared, for example, here on 3B that was not there at seedling stage, and that could be an adult plant resistant gene. So here there is what I'm showing you in this graph is based on CASP marker, the competitive allele specific PCR marker that are diagnostic for this known leaf rust resistant gene in hard winter wheat. You can see there the frequency of these resistant genes like LR21, 24, 37. They are present, but they are not effective. So we know that whatever we have here are different from the known resistant gene in hard red winter wheat. The APR, we have some like common resistant genes like LR34, 46, 67, and 77. So Indira now she's looking at the gene or the associations that are novel, that are distinct from the ones that we already know in hard red winter wheat. And she's developing CASP markers, competitive allele specific PCR markers. Those can be diagnostic marker for these novel genes or genes that are underutilized in the breeding program, and that's to assist with selection in breeding. Rajat is a PhD student who is working on stripe rust resistance. So he is working with the USDA in Kansas and Washington to do the evaluation both at seedling stage to these different races of stripe rust pathogens. So PSTB37, this is the most common race that we have in Oklahoma and in the US. But these races for 14, 40, and 52, 52 can be present also in other locations, but they are present in Washington. So those are originated from Washington State, where the environment is more conducive for the stripe rust, and also we have higher diversity of the pathogen in Washington State. So this is the infection type scale that he's using from 0 to 9. You see that we have a few number of lines that were resistant, but we have high number of lines that are susceptible. So the number of lines that are carrying seedling resistant gene in this germplasm of hard winter wheat is low. But by looking at the resistance in the field, and we have 3 locations. We have in Pullman, Washington. We have an experiment. We also have in Mount Vernon, that's in western part of the state. We don't have the data yet. This is a 2023 data. And for Kansas, we have a nursery in Roseville in collaboration with the USDA. And we have a nursery in Oklahoma in Chickasha. So we have the infection type, which is 0 to 9, and severity is 0 to 100%. And you can see that we have here more lines that are resistant. So this shows that we have adult plant resistant genes in hard winter wheat that we don't know where they are. And that's the research project of Rajat. We had a visiting scholar from Egypt. Just like from May, I believe, to June. She just left, or no, to July. And she helped with the stripe RAS evaluation, and this is when she was helping us in Pullman to take the phenotypic data in Washington. So Rajat, he did the analysis for all the data, but due to the limitation of time, I'm showing you here just some example of his Manhattan plots using the B-Link model. So the first graph is for PSTV37. So have primarily a loci on 1B, on 2B, and on 4A. And by looking at the known stripe RAS resistant genes that are expressed at seedling stage, like YR17, that is present in high frequency, it is not effective against PSTV37. So we know that we are identifying other sources. They can't be YR5 and 15 because of this low frequency that are present in the population. So we know that we are identifying some even novel or underutilized stripe RAS resistant genes in the US hard winter wheat breeding programs. These 2 graphs, those corresponds to field data in Roseville in Kansas and in Pullman in Washington state. So more associations, and that is expected because we were seeing higher resistance level in the field. And what we know for stripe RAS are only 2 genes for adult plant resistance, YR18 and YR29 to be present in high frequency based on diagnostic marker. So we know that we are identifying other associations. So, and also here by looking at the different locations. So this is the reason why we put these nurseries in different locations, because we have different environment and also different races that can be present in these locations. So that's why we can see some common, but also some distinct associations like this one on chromosome 6A. So with that, I came to the end of this presentation, and I would like to acknowledge the people who did the work. The OSU wheat improvement team, primarily Dr. Brett Carver, who is the wheat breeder at OSU. Our collaborators from the USDA, Dr. Colmer, he's from USDA Minnesota, for his help with the leaf RAS project, Dr. Chen and Wang from USDA Washington for their helping us to set the stripe RAS nursery and with the evaluation at seedling stage, Dr. Bob Bowden and from USDA Kansas for putting our nursery in Roseville in Kansas, Dr. Bai Ahmed and Bernardo for the genotyping at the USDA Kansas, and Dr. Jeff Baum from USDA Nebraska for distributing the seeds of the regional nurseries. Of course, my funding sources, USDA NIFA and the Oklahoma Wheat Commission, and my lab, Brian, he's the technician who is doing most of the work that I showed for the OSU breeding program. Indira and Rajat, my graduate student. Finn, he's an undergrad helping with everything in the lab. Dr. Hamza, who came from Egypt to help us with the stripe RAS project. And Maria, who is an intern from Ecuador, who helped with developing a diagnostic tool for viruses. So please feel free to contact me. This is my social media or my email. So feel free to contact me if you have any questions. And we'll leave you here with this nice picture from our OSU football photo. So thank you for listening, and I'll be happy to take any questions that you may have.
Okay, thank you, Dr. Meriem, for this really interesting lecture and good research for wheat rust resistance. And now we open discussion, so you can write your question in the chat and I will tell to Dr. Meriem, or you can raise your hand and I will unmute your mic. So, yeah, so we have a couple of questions, Dr. Meriem. The first one in the chat, so you can read it as well.
Yes.
For developing fungal disease resistant wheat variety, do you use conventional breeding method?
Yeah, so for developing resistant variety, we do the phenotypic evaluation. That's what we call conventional breeding, but we also rely on molecular markers that are available for diagnostic marker, like diagnostic markers, Cas markers that we know for specific resistant genes. So if we have them, then we do the selection based on molecular markers and also based on phenotypic data.
The second one, no?
Yeah, you mentioned that you use high throughput phenotyping methods. Do you please give me some example of these methods? Yeah, so we start doing like flying drones. So we are using hyperspectral camera and also multispectral camera for phenotyping of barley yellow dwarf. So we are collecting the images. So we still have to process the data and see how we can correlate the data with our phenotyping and see if we can basically develop this method for breeding, because we do annually like thousands of breeding lines that we test in the nursery. So if we can have something faster, that instead of doing phenotyping for days, maybe we can do it in 15 minutes by flying a drone.
Okay, we have Danielle. Please unmute your mic. You can speak. Danielle, you can unmute your mic, please. Okay, we have Isa. You can unmute your mic, Isa.
Can you hear me?
Yes, I can.
Yeah.
I have a question for Dr. Meriem. You mentioned that your master's student, Indra, I think on that slide you mentioned she got only one association in the Manhattan plot by form CPU. So my question is, why you don't prefer the mixed linear model for JWAS?
Yeah.
As it gives more prediction accuracy than form CPU.
Yeah, so these are different models that are developed, and the community now, you can use mixed linear model. That's like we use different models. We use mixed linear model, B-Link, Farm CPU, so many other models, but we selected the best model based on the QQ plot and also Farm CPU. So it's a multi-locus approach. So it has it is a more powerful approach. Or a more powerful model to use, B-Link and Farm CPU, and also computationally they are faster. And it is really helpful, I think, for me, like when I use mixed linear model, I will get like several associations, but all in the same locus. So with Farm CPU, it just limit that association to a single locus. And in recent publications, you will see that more of this multi-locus models are being used, like Farm CPU or B-Link. So that's why we are testing different models, but we are selecting the best.
Okay, thank you.
Yeah, sure.
Okay, so we have a question in the chat room.
Daniel?
Yeah.
GWAS analysis normally leads to identification of many genes. Could you please explain to me how you select the putative genes? Yeah, so it's basically the GWAS. It has a higher resolution. So we can look at putative genes, but basically I won't really, you will have so many genes in that region. So we don't look at specific genes, but we know that this locus or that marker, if we validate it, then we can use it in selection. We are not necessarily going to get what gene is involved, although GWAS can be used also for cloning genes. So if we get lucky, maybe in getting some genes that are associated with resistance. For example, if we are looking at seedling resistance, probably we are look at the MBLLR genes that are present in that area in the region, but if we are targeting adult plant resistant genes that are different than the MBLLR genes, so we have, it's like the situation is more difficult. So we if we go for a specific marker, we go with a validation in a different population before we can use it in breeding. But we are not necessarily going for gene, but we are targeting markers that can associate it with the gene. Okay. Next question. Synthetic wheat lines are valuable resource for resistance to diseases. Do you have checked or planning on synthetic wheat lines in your breeding program in the US? Yes, that's something we do for viruses because we are having this resistance in wide relatives like in tinopyrum, and we are working with Kansas with the Wheat Genetic Research Center in Kansas, that they already developed synthetic wheat where they get the resistance source in synthetic wheat, and that will be easier for breeding rather than just going to the wide relatives. So yes, that's something, and I actually have in my office some synthetic wheat from them that we are using for breeding. Okay, I see another question.
Yes.
Do you look for more details on the possible new genes controlling YR seedling resistant gene? For example, gene models harbor the significant markers, their functional annotation. Yeah, that's something interesting to look at. But we basically at this stage we are like looking at this association and see whether, based on the molecular markers, whether and the physical location of these associations, whether they are present close to known resistant genes. So that's how we claim whether they are a new region, or like it's a new association, or it is already known. And we don't necessarily look for new genes, like even genes that are known in wheat, but they are not highly utilized in hard winter wheat. That's something that we tend to develop a marker. And even it's a known gene, but it doesn't have a diagnostic marker. So our work with the GWAS that hopefully is going to give us a higher resolution will help us to develop these diagnostic markers. But that's something, yeah, we can look at. But that's not something we are doing actually.
Okay, continue, please. Going again to the high.
Going again to the high throughput phenotyping methods. The types of camera you mentioned are using only for viral diseases, not for fungal, or could be used. Yeah, it can be used for any disease. Not only for disease. Like we are using actually this high throughput phenotyping for yield, for height, for other agronomic, but we start doing it for viruses. But if we can use it for other diseases, especially if they are difficult to rate, or they have this quantitative type of resistance, like I would probably see it to be very valuable, for example, for FHB, Fusarium head blight.
Okay, we have a question from Dr. Dua Hamza. So please, Dr. Dua, unmute your mic.
Hi, Dr. Meriem.
Hi again.
How are you?
I'm good.
Thank you so much. I just want to thank you for everything you do with me there. You are very helpful and supportive to me in all my steps there since I arrived to US to till I'm travel. You are amazing.
Thank you.
One.
No, no,
life.
thank you for helping us with the program. Actually, we appreciate your visit and your hard work.
Thank you so much, Dr. Meriem, and I wish we will meet again.
I hope that too.
Okay,
Okay,
thank you, Dr. Ahmed, for your presentation and for your invitation to Dr. Meriem to give us a very amazing lecture.
Yeah, thank you, Dr. Dua.
Thank you.
Okay, we have.
Daniel?
Do you have a question, Dr. Dua, or do you have a question, Dr. Dua, or?
I'm just thankful to Dr. Meriem.
Okay, thank you,
welcome
Dua. Thank you.
Coming again to Dr. Meriem for the chat. Did you sequence the tolerance lines that are phenotypically identified to identify
No,
if there is? No.
We don't do tolerant lines. The tolerant lines that are phenotypically identified to identify if they are any indels or not. We don't. Just we rely on the SNP markers that we have, but yeah, that's something we can consider. But we didn't.
We have another question.
I didn't notice if you showed how to treat wheat with RAS spore in the field to record phenotypic data. Please, can you explain that? Yeah, I didn't show how we do it, but it's just simple. We get spores. Of course, they should be spores from Oklahoma. We don't want to use spores from other states or other country to do inoculation in the field, and then we mix them with Saltrol. Or we can also put it in water with some twin and then basically do the spray in the field. And we spray only the spreader rows. So those are susceptible ranges that we put in the field, and that's to help with the infection, or the border rows. So we just do spray that mix of Saltrol with the spores, or it can be simply water and twin 20. We add it, and then we do the inoculation. And basically, after the leaves dry, we are targeting days where we can have dew. So if we have good moisture, that will help with the infection. Otherwise, we try to irrigate, like do a mist on the leaves to help with the infection, and that will be in the evening. Another question. Would appreciate to be in touch in your team are involved in breeding for abiotic stress. Yeah, that's actually will be, I believe, Dr. Carver, who is the wheat breeder, and I think you can find his information on online. Yeah, he's the one working, basically try to have something for drought, especially that in 2022 and 2023 we had severe drought, and also with heat tolerance too. We are having unexpected like high temperature, but here in Oklahoma, we basically our crop will end in June. That's the latest before we do the harvest. So we definitely with the drought that we experienced this last 2 years, 2022 and 2023, that's something important to have in breeding.
Okay, we have another question from Yusuf.
Does most infection occur in the adult plant? So this study's concern was on adult plant resistance. No, it's not because the infection occurs at adult plant stage. No, we are doing seedling and adult plant stage test, and that's to distinguish between the resistance, the seedling resistant gene versus the adult plant resistant genes. So I think if I got your question right, so we do the test is just to distinguish between these two types of resistance. And basically at adult plants is just one, maybe at flag leaf stage, and at seedling stage, the rating we do it like at second leaf stage. That's when we do the evaluation. It's just to distinguish between these two types of resistance. And adult plant resistant genes are more durable. So that's why we are targeting adult plant resistant genes.
Again, from the same person.
The sprinklers. That sprinklers irrigation reduce the infection. How is that? No, we try to do like, we don't irrigate like most of the time. We don't irrigate. We basically target a day where we have like, let's say on the weather forecast, it says that we will have dew tonight or rain. So we basically go and inoculate, and then at night we'll have that rain, and we need that moisture, that humidity to help with the infection. If we don't have it, then we try to, try to have something that looks like a very light rain.
So do we have further question to Dr. Meriem or? Okay, I have a question. I can say now my questions.
Sure.
I would like to discuss the GWAS results regarding to your study about disease resistance. That I think it is very important also to consider the markers that you find in the GWAS results, because I have seen in your Manhattan plot that you have like high peak of some good markers. So I would like to ask you, did you find a common marker across years that appeared to associate with like yellow rust resistance or strap rust resistance, so can be used for a selection in future breeding program? Because I have understood from your presentation that you relied on the known genes for stripe rust and leaf rust resistance. So, but it could be GWAS providing new sources of marker, and you can also select the markers that have high R square, phenotypic variation explained by marker R square. So it's like something like markers explain more than like 20 and 30 percent of phenotypic variation. So this marker could be useful source for resistance.
Yes.
We have experiences on this when I was working in US in Nebraska breeding program, that we always select the markers that had high R square, and then we looked for the genotypes, how many markers in the selected genotypes, because we relied on the phenotypic selection and selection based on SNPs detected by GWAS. So did you do something like that, or especially the common marker that appeared significantly in more than 1 year or more than one locations?
Yes. Yeah, thank you for sharing your experience. Actually, that's what we do also. So we look at the shared markers from the different locations, also for the shared markers between the different races. And we are basically trying to target primarily the ones that have major effect. So we are doing an additional to what we have, like the R square and everything. We are doing a t-test between the two like homozygotes group, and select the one that has also a significant p-value. We are targeting the ones that have, because we don't want something that has a minor effect. It's like a breeder, they won't use it. So we want something that can be of value for the breeding program. So that's something we are focusing on. And I like the graph that I was showing based on the known genes. Actually, that's to show that we have other association than the known genes. So diagnostic marker, that's fine, but also we want to find something new based on the new association that we have. So yeah, we are doing what you have experienced. So definitely having these major effect markers is our priority for CASP marker development.
Yeah, actually, yes. Anyone has a question to Dr. Meriem? So I will check in the chat room. Yeah, we have.
What role of plant tissue culture to avoid the infection of this disease? We don't do any tissue culture for this. Like basically that's only if we are doing like transformation, for example, to validate some gene with like, you know, silence gene or overexpressing gene. That's when we do, and that's like collaboration with Dr. Yan, who is like an expert in gene cloning in our weight improvement team. So we don't do any tissue culture unless it's for, you know, doing something like to do mutation or like to, if we, that's like, you know, basically that's part of validating if we have to clone a gene. But at this point, no, we don't do that.
Okay, so I know that we a lot of questions with this lecture. So do we have further questions? Okay, so I think this is the end. So if anyone has a questions, we have the email of Dr. Meriem on your email or in our websites. You can freely contact her in any time. And I would like to thank you again, Dr. Meriem, for this outstanding presentation. We really get a benefit from this presentation, and we hope to see you again, maybe next seminars. To share new knowledge to us about wheat rust resistance in US.
Thank you for listening, and thank you all for staying this late to listen to my seminar. I appreciate it.
Okay, thank you, Dr. Meriem.
Bye-bye.
Bye-bye. This is the last MGBS this semester, and regarding to certificate of attendance, we will send you an email how to get it. We will allow the link of registration for more 15 minutes if you would like to register or if you didn't register yet. And thank you for all the attendees of this lecture and all previous lecture, and see you in our next event. You can follow our next events in the website or by Facebook, LinkedIn. We will copy paste all links of our upcoming event. Thank you all, and see you next events.